Hi, everyone. Thank you so much for joining us tonight. We have a really exciting presentation planned. This is going to be Saltshakers of Death, researching the fungus that turns cicadas into zombies. Dr. Matt Kasson, who's an associate professor of forest pathology and mycology at West Virginia University, is going to be speaking with us tonight. And before we get into the talk, I do just want to put out a plug for a fungal plug, if you will. I'm so sorry. The if you're not a member of the IMA, but you're here on the call because this sounded like a cool subject that you wanted to learn more about, you're correct. Good. We're happy to have you. And I wanted to let you know we have two surveys coming up. One of my favorite things that the Illinois Mycological Association does is we go out and collect research surveys of fungi in the forests of Illinois. And only IMA members can come on these surveys. So if you are not yet a member but would like to be, you can go to our website, which is illinoismyco.org, and sign up, become a member, and then you'll find out about all these sorts of talks in the future, and you'll be able to come to our surveys. We have one coming up on June 15th and one coming up on June 29th. So those should be tons of fun. Definitely please come join us if you haven't, if you're not a club member yet, or even if you are, just haven't been in a minute. I also want to plug our group survey chair, Liz Weinstein, is giving a presentation about edible mushrooms next week on Wednesday, June 12th at the Downers Grove Public Library. So Liz is really cool. That should be a really fun talk. Everyone should come if they can. So with that, let's get into some zombie cicadas, shall we? I am thrilled tonight to introduce you to Dr. Matt Kasson. He's actually spoken to the IMA before, I think, but and never in a more timely moment than this, right in the middle of the big double cicada brood emergence in Illinois. Dr. Kasson received his Ph.D. in plant pathology from the Pennsylvania State University, where his research focused on using a native fungus as a biological control on the invasive tree of heaven. He also holds an A.A.S. from Paul Smith's College and a B.S. and M.S. from the University of Maine. His current research areas include fungal-arthropod interactions, biological control of invasive plants and pathogens, and the biology and ecology of historic and emerging diseases of forest trees. Dr. Kasson teaches undergraduate courses on general plant pathology and forest pest management and offers special topics courses for graduate students, including advanced plant disease diagnostics. Without any further ado, Dr. Matt Kasson.
Thank you, Kate. Thanks for that nice introduction. I'm excited to be here for a number of reasons. One of them being that I love zombie cicadas. The other being that I'm coming to Chicago for the first time. I've flown through Chicago, but I've never actually been to Chicago. So I'm excited about that. So I'll have to get some tips and things about must eat places, although I'll probably be too busy collecting cicadas to actually do anything else. As you all know, I'm going to be talking about the Saltshakers of Death, the zombie cicadas, and really what this dual emergence can teach us. Just a shout out to my former student, Molly Sherlock, who drew this Massospora infected cicada. And we have stickers available if I run into any of you in during the next few days when I'm in town. So I think I'll start by talking a little bit about cicadas. It's kind of implied that a lot of people in this group know about fungi and kingdom fungi. Oops, I apologize for that. But fewer of you may know about cicadas beyond the periodical cicadas that are just screaming in your ear all around us. But cicadas, they're in the superfamily. They're a superfamily in the order Hemiptera. They have piercing, sucking mouth parts, so they feed on sap. And they're closely related to plant and leaf hoppers. One notorious plant hopper we have here in the East is a spotted lanternfly, which is a visitor from far away, and we don't like it. But when you get up close to those, you see the kind of cicada-like resemblance and their kind of awkward flight patterns. Now, regarding cicadas, there's some 3,000 species worldwide. Of course, that's probably a gross underestimation because, biodiversity discovery being what it is. There's a lot of undiscovered cicadas. But most of the cicadas, whether they're known or not, whether they have a Latin binomial or not, a majority of them occur in the tropics. Now, we are being inundated, or you are being inundated right now by a 17-year cicada brood, and that is brood 13, and that is a periodical cicada. Periodical cicadas spend either almost 13 or almost 17 years underground before emerging, and then having that short few weeks of adulthood where they mate and pass on their genetic material to ensure the next generation. Contrary to popular belief, annual cicadas don't have a 1-year life cycle. It's just that some of the population, some individuals emerge every year. So annual cicadas still take a couple of years underground to develop as well, like the dog day cicadas we hear and seldom see in the hottest days of summer. Those are annual cicadas. I'll have to apologize because the bar for Zoom is kind of over some of my slides, so I sometimes have to shift it so I can see everything. And regarding the periodical cicadas, we know that there's 7 Magicicada species, and that includes 4 13-year cicadas and 3 17-year cicadas. There's also 15 extant broods. That's 3 13-year cicada broods and 12 17-year cicada broods. Now, these broods occur in different time and space, and I'm sure with all the articles written, including articles like Kate wrote, you get a little bit of clarification about where the footprint of these different broods occur, when they're coming back, and things like that. This is a nice figure from Chris Simon's annual review, kind of showing the larger footprint of 13-year and 17-year. And then showing the individual kind of fingerprints. So you can clearly see there's some overlap in the broods, but what you can really see is that the 13-year cicadas are mostly confined to the deep southeastern United States. And the other thing that we should bring up is that these broods are expanding and contracting based on what's happening. We know that weather patterns are obviously important, but fragmentation of the landscape, seldom do we think about the cicadas that have been underfoot for 16.9 years. And in those 17 years that they spend, or mostly 17 years they spend underground, trees may be cut down, developments may be put in, and those cicadas are effectively erased from the landscape because they are sac-feeding insects that in their nymph stage are feeding on roots. And if you kill the roots, you kill the cicadas that rely on them. So as such, these broods have expanded and contracted over time. And even brood 13, which is in your neighborhood right now, is a relatively small brood. And the question is, when we start to have more extreme weather or some other things, tip the scales in favor of things other than the cicadas, at what point will they no longer have a viable population? I hope we don't see that, but it's something we need to be thinking about even when they're underground. Now, the cicadas themselves, true cicadas have 2 families and 3 subfamilies. This is not a comprehensive phylogeny, but one to show kind of the relationships among some of the cicadas, with some red arrows indicating some species that I'll be talking about today. There's Okanagana, which is an annual cicada we see out in the Midwest. There's Platypedia, which is a wingbanger cicada, an annual cicada we see out in the Southwest, including California. And then Magicicadas up here in the pink. So you can kind of see where they fall relative to each other and kind of what subfamilies they fall into. And I think, a lot of us that work with, whole groups of organisms, we're often thinking about the relationships among those individual species to try to understand susceptibility and patterns that we're seeing across the landscape. Now, cicadas also have some really interesting quirks. And I'm bringing them up not because I'm trying to fill time. Um, I am doing that, but I think it's important to understand that there are mutualistic relationships happening with cicadas in the absence of fungal pathogens. And these types of relationships are important and we'll come back to when we start to think about what's happening underground with Massospora and what's happening with these nymphs, given what happens with them, normally. So we know that they have these piercing, sucking mouth parts, and I'm sure there's plenty of entomologists around, maybe even on the call, that can explain kind of, how they count sap cycles to know when to come up together to synchronize. Um, they have obligate bacterial endosymbionts that are stored in specialized kind of organs in their abdomen or in their gut called a bacteriome. And these provide essential amino acids to supplement their diet because they are sap feeding insects. And sap is not nutritionally robust. It's a somewhat nutritionally poor substrate. And the quality of sap, the nutrition goes down, based on a kind of an annual cycle. So being able to supplement it with an internal bacteriome and these bacteria that can provide essential amino acids is important. It's been discovered recently in recent years, including John McCutcheon's group, that a certain number of cicadas have domesticated Ophiocordyceps. And those on the call that are fascinated with insect pathogens will quickly recognize Ophiocordyceps as, among other things, a zombie ant fungus. And we'll talk a little bit more about summit diseases here in a moment. But some cicadas have basically domesticated Ophiocordyceps. And those Ophiocordyceps have in turn supplanted those bacterial partners and are now providing nutrition in the guts of these cicadas without needing any kind of bacteria in there. So it's kind of a wild situation. And we'll venture back to that concept a little later on. The other thing that we have to understand is, the kind of courtship that happens between, periodical cicadas, for example. Not all cicadas vocalize. In fact, one of the cicadas we studied, the wingbanger cicadas, communicate only through wing taps. But we know these periodical cicadas are not like that because they scream. And it's really just the males that are screaming, right? They have the tymbals, those specialized, organs on the back of their last leg pair, and their abdomen serves as kind of a resonating chamber. And they can really throw out some sound, as you all know by now. And that's important for, impressing the ladies. And then if the female cicada is receptive to that call, she'll flick her wings, and that wing flick will say, I am available for a mating attempt. I am receptive to a mating attempt. And that's an important interaction that happens between healthy cicadas. And we'll talk about the different versions of that as we get more into Massospora. Now, of course, there's a lot of different fungi that can invade and colonize cicadas. There's Ophiocordyceps that can infect, nymphs. We've seen this in citrus cicadas, Disroprocta cicadas out west. They've studied and described a lot of these from Japan and elsewhere. There's Buvaria, there's Metarhizium. In fact, I know a number of cicadas I've seen, show up on iNaturalist or just show up in the mail in a package to me. Will have co-infections. You'll have a Massospora infection in the back, and then you'll have a Buvaria infection, kind of like you see here. But eventually, if it's a Metarhizium, you'll start to see them green up because the Metarhizium, has that kind of green, kind of dry spores. So these you can call this Buvaria now too if you wanted to, because it looks very much like that as well. But these are important players, but not quite as cool and as important as the zombie cicada fungus Massospora. So what do we know about Massospora? Well, if you go to the Wikipedia page, there's reports about anecdotal reports from the 1840s and earlier about abdomen sloughing off, but it really wasn't until Peck formally described Massospora cicadina from a brood 2 periodical cicada in New York that was collected in 1877. Peck wasn't sure where to put Massospora cicadina, but Roland Thaxter came along 2 years later, or no, maybe a little while later, 1888, said, no, this is an Entomopteroides. And sure enough, that's where it's sat ever since, although the phylum has changed. Massospora is a group of obligate sexually transmissible species that infect some 21 cicadas worldwide. And I'll show you a list here in a second on the next slide. But after its formal description and kind of, after Peck and Thaxter did their work, it was kind of a mycological oddity for 100 years. There were some kind of, a few papers that came out, one in 29, I think, but it was really, it wasn't until Richard Soper came along and in the first issue of Mycotaxon formally described all these different Massospora species, recognizing basically all the genera, with the exception of one or two, which he described later on in the 80s, describing basically all the Massospora species that we know. And here's to kind of show you the diversity here. Here's Massospora cicadina here, but you can see Massospora levospora and Platypedia, which we'll talk about how they're basically synonymized. Massospora dysrhoprocta is kind of unique. It produces a purple plug on the back, but sure enough, it is Massospora. And this is Massospora tetigades from some of these hairy-looking cicadas in Chile. And we'll talk a little bit more about some of these things. Now, you can probably imagine why it was a mycological oddity for 100 years. If you had to build your career around something that emerged once every 17 years on a subset of cicadas, and we didn't have an interstate road system or a quick way to get around and to do windshield surveys or whatever you have you, it'd be really hard to make a living off studying Massospora. And that's why it remained a mycological oddity. And then in 2016, my research group picked it up because they emerged on campus. And, when opportunity comes knocking, sometimes you have to pick up. So here's a list of some of those described Massospora species. I have some highlighted in darker black. Those are the ones that really our early work that we published on in 2020. This was led by my graduate student, Angie Macias. And we basically had specimens from 5 different species, including tetigades from Chile, Platypedia from out west, California, New Mexico, levospora from, we had some from Michigan, but they're kind of spread out. Dysrhoprocta, we had some from Arizona, and cicadina, of course, we had the most of, and then we had hundreds of samples. But thanks to some supplemental specimens from Gene Kritsky and Chris Simon and other cicada biologists, we were able to kind of move forward with kind of a first robust, comprehensive study of this group of enigmatic fungi. Interestingly enough, when you look at the first deposited, sequences, and I think it was, 28S and ITS, it's kind of a hint that it was an ITS sequence, but the first deposited sequences for Massospora were actually Entomophthora musci, and it was a number, it was a clerical error, because RCEP 3074 is Entomophthora musci and RCEP 374 is Massospora cicadina. So when we generated our first sequences out of our own specimens, they did not match the one that was deposited. And only through phylogenetics did we realize that, sure enough, it was an Entomophthora that was originally deposited. But we were able to go back to the actual physical specimen and re-extract DNA and confirm that it was Massospora. So you can see kind of a phylogeny here down on the bottom, just showing the relationships among these Massospora we looked at, and I kind of have them color coded up top, so you can kind of see the cicada, but also see the where they fall out phylogenetically. And you can see cicadina here is kind of forms, is sister to or forms a clade with Massospora dysrhoprocta. And then you have basically a single clade that comprises both Massospora levospora and Massospora platypedia. And that was really interesting because in that situation, the cicadas don't overlap, those hosts, Platypedia and Oconagana. There are Oconagana species in California, but the specimens we had from Michigan and then the specimens from New Mexico were in kind of completely different geographic areas. But we generated sequence data enough to show that, okay, well, of the 5 species we had, we basically confirmed that there was 4 genealogically exclusive, well-supported clades, suggesting that there was 4 species, not 5, based on the data we had. So it was fun to generate that first phylogeny for Massospora, but certainly not our last, and we're hoping to generate more soon. Of course, because Massospora was this mycological oddity, that means that the sequence data was already limited, even if it was wrong. So the only way to really link to these historic studies is to do this kind of classical morphology work. And my lab is really takes that kind of role very seriously to balance, to do those complementary things where we look at morphology, spore measurements, and other metrics, and then couple that with kind of the more molecular-based investigations. And what this work showed for these 5 species is that the mean conidial widths differed among all species included, but there was a lot of overlap in the conidial lengths, and you can kind of see that here as indicated by the significance letters. So interestingly, I've already showed you the phylogeny for and told you that Platypedia and Levospora are basically synonymous. So ML and MP are positions number 3 and positions number 4. So you can see based on resting spores, they're not significantly different, but based on conidia or stage 1 spores, they are different. So if you're using morphology prior to PCR, you can see why you might have called them 2 different things. They were on 2 separate hosts. They had different, spore sizes, so that makes sense. But what we're learning is with these kind of strange, co-evolved, specialized fungal parasites. Sometimes you can find morphological differences, but it may not be as meaningful as in other cases. But of course, there's always barriers. The biggest being that, despite attempts even by us to get this going, it can't be cultured as of yet. That's not to say that we can't crack that code, but we've tried Grace's media, we've tried breakfast media, cicada wings. We boiled, 400 cicadas and made almost like a slurry, and then poured it through cheesecloth, and we had cicada extract auger. Didn't work. It's also not really a system conducive for librarian. Let's get a lab colony going. I know 17 years is a long time to wait for your PhD, but it's going to be worth it. And then year 15, your colony dies out. It's just not a practical system to study. And as such, we have to really rely on field collected specimens that we can archive, that we can deep freeze, that we can tease apart. We have to opportunistically collect when we can. There's also this incomplete understanding of the disease cycle. This was a figure that my former postdoc, Brian Lovin, and I put together for 2020 plus pathogens. Pearl we put together on Massospora. And, it's an interesting figure. It kind of shows what's happening. It goes from conidial plug to resting spores. But there's a lot of question marks. First question is, in the earliest cicadas that have stage 1 spores, if they infect another cicada, do they too get a stage 1 spore plug? Or does it always go from first contact stage 1 to second, stage 2 and the second cicada? We don't know. Another question, and the biggest question of all, is what happens with those spores at the end of the season? One of the things that I will come back to several times is that we've put these spores, the resting spores, on standard growth media and got them to germinate. Now, they don't continue to grow beyond that. But what that tells me is that most of these spores would mistakenly germinate before they ever encountered a cicada 17 years later. So how do they still maintain enough inoculum to infect enough cicadas so that they too can persist into the next generation? And really, I think it comes down to what happens on the way down. We'll come back to this point, but one thing I want you to start you thinking about and get your kind of creative juices flowing about is that we are now starting to think that infection happens in nymphs before they even get, or as they get into the soil on their way down, and they persist with the infection, maybe in a latent period or maybe something else, until a cue like a molting hormone or something allows it to transform from a neutral or beneficial stage, beneficial relationship to one of parasitic. And we can certainly discuss that more if there's questions at the end, but I'll kind of recapitulate and talk a little bit more about here in a second. So much of what happens in kingdom fungi happens below our feet. And we take that for granted. That's true of Massospora too. So much is happening under there. For example, maybe a certain percentage of nymphs do get outward infections and it does explode out of them. And we never see them because they never surface. There's so many that die under the ground, but we don't account for them because there's so many that still make it up. Imagine if they all made it up, how many more there would be. There's just a lot of questions, but it's exciting. It's an exciting time to be working on this. And just understanding where the fungus goes, how it gets in, what's it doing, all that. So one of the most fascinating things, and the reason that I get asked to do interviews all the time is not because of the phylogenetic tree or the resting spore ornamentation. No, it's the behavioral modification, the extended phenotype of the fungus. There's actually two distinct behavioral modifications that happen with Massospora. There's something called active host transmission, and this is something we wrote about. My former postdoc, pictured here, Brian Lovett, who's now a insect pathologist at USD A.A.S. In Ithaca. And active host transmission is not unique to Massospora, but it's popular now because of Massospora. There's also another fungus called Strongwellsia, and I'll get into another example here in a second. But what basically, you're all familiar with zombie ant fungus, Ophiocordyceps. That's a summit disease pathogen. So what happens there? A fungus infects an ant. It coerces the ant to ascend to a high point, affix to a substrate, bite down or latch on, after which the fungus erupts brilliantly from the cadaver post-mortem, and then spores are raining down onto unsuspecting ants below. A lot of insects have this kind of summit disease type of transmission. Active host transmission says, no, you know what? I want to keep my host alive. And it makes so much sense for a 17-year cicada. It makes sense for other cicadas, but it makes a lot of sense for 13 and 17-year cicadas. You have co-evolved alongside cicadas for millions of years, and you have to make sure that you too survive. You're not going to bring your cicada host away from this screaming mass, this super spreader event. You're going to participate in it, and you're going to ensure that you maximize dispersal. And that's what active host transmission does. It is a modification that benefits and optimizes spore dispersal over the wishes and desires of the host. And it is the fungus that is the puppet master pulling those strings on the cicada to modify its behavior to maximize that dispersal. There is another behavior, and this is the hypersexual behavior that I think every article that's ever been written on it really likes to celebrate. And this has been shown that males, for example, that have infections, in addition to screaming at lady cicadas on close by branches and hoping that they flick their wings and, to indicate their receptiveness to a mating attempt, the males will also pretend to be females and flick their wings in the presence of another screaming male. Again, this makes sense from, an extended phenotype kind of puppet master standpoint. The fungus wants to optimize dispersal. If you're only spreading among half the population, are you going to maximize dispersal? Heck no. You want to get in contact with as many cicadas as possible. And once the cicadas get close, once that like wing flick happens, kind of back into each other. They have a couple ways that they attach, but they're just kind of they don't check. So they back into this fungal plug, and sure enough, now they have the salt shaker of death that's come in contact with their genitalia, and they're doomed. This grotesque picture you see here in the center is one that shows a male that still has the genitalia and ovipositor of its female partner attached. What happened here is that the female was infected, but because they have such strong muscle tissue surrounding the ovipositor, the abdominal sclerites don't often fall off as easily on the females. So the only way that break happens is if they oviposit in a branch, or if they try to mate. And as they're separating, the male will then break off the female's posterior abdomen, genitalia, and ovipositor, and fly around with it attached to his genitalia. Pretty grotesque, but hey, this is a stranger than fiction story, and you're not here for something boring. Now, I mentioned that active host transmission was not specific to Massospora, but Massospora and Strongwellsia are definitely known for that. Strongwellsia is another bizarre one. Check out this fly here in the lower left. You can see a perfectly round circle. And I don't know if that's a combination of enzymatic attack that allows for kind of active spore discharge, because the spores are actually shot out of this hole. Whereas on Massospora, it's more, there is that salt shaker of death where the spores, the cicada is flying around and spores fall off like chalk dust. But on the fly with Strongwellsia, they're actually actively discharged off of like specialized conidiophores. There's a couple of like Entomoptera cansana, for example, that's been described. And then there's this Entomoptera abrupta on black grass bug or the green apple bug. You can see that here. It's really interesting. You look at this one too, just like Massospora, you're like, how is that insect still alive? But sure enough, you could have debates on insects, pain receptors, and what these fungi are doing for the insects. But I think what you can see is that it's clearly having an effect on the outward appearance of the insects. Regarding Entomoptera, these are probably not really Entomoptera. Everything was kind of grouped into Entomoptera because it was kind of type genus for the family. These are probably something else. I've gone on social media before and offered $150 a sample for this, and I've never got one. If you find one, I'll pay you for it. Same thing with a Massospora infected dog day cicada. There's a picture of one on a publication from the 90s, and but I've never seen a specimen in a museum or an herbarium, so be on the lookout. So what can describe this behavioral modification? Of course, if you've studied fungi long enough, or you read about fungi long enough, you know that they're brilliant secondary metabolite producers. They have diverse biosynthetic gene clusters. We give them credit for lysergic acid and ergot alkaloids. They produce penicillin. They produce really interesting immunosuppressant drugs that are critical for organ transplants. So I had a student who was doing global metabolomics of arbuscular mycorrhizal fungi at the time, and I had a bunch of Massospora spores. And I said, we should look at these and see if we can find anything interesting in these fungus-infected cicada plugs, or these fungal plugs from cicadas. And we had both periodical and annual cicadas. And I'll never forget, as long as they live, the call that came in, like this was back in 2017. It was like 2:30 in the morning, and I get this call. My cell phone was ringing. my wife's like, who's calling you at 2 a.m? I'm like, I don't know what's going on. And it's Greg Boyce, my PhD student. And he said, you're not going to believe this. You're not going to believe this. He's like, I was processing some of the metabolomics data, and I found psilocybin in all of our wingbanger cicadas from out west. And I was like, are you sure? He's like, well, there's a lot of things we have to do. But I'll talk to you more in the morning about it. And, we eventually met up, and I asked him, is there psilocybin in periodical cicada plugs? And he looked, and no. But then something jumped out. There was this really high ranked, compound, and it was called cathinone. And I had never heard of it before, but this is a stimulant found in cat plant and an amphetamine. And sure enough, it was in those plugs and not in the healthy cicada abdomens that we used as controls. And fortunately for us, these compounds have DEA exempt analytical standards so that we can kind of compare the direct fingerprint of these compounds with what we found in the plugs. And it's only through that, comparative analysis that you can not only confirm within a certain ppm that it is in fact the same fingerprint, but then you can actually do quantification and ask and answer the question, well, how much of that stuff is in a single plug, and what's the biological relevance of that? So really interesting that, in a matter of a couple weeks, we found what appeared to be an independent evolution of psilocybin production in kingdom fungi, and the first evidence of amphetamine production in kingdom fungi. And that was a bit of a wild ride. Because we had the analytical standards, we were able to quantify it. And you can kind of see the cathinone in nanograms per fungal plug here in this little bar graph. Sorry about that. So cathinone wasn't present in a lot of the plugs. It was present in 32% of the plugs. And it was only quantifiable that it was above a certain threshold in about 24% of the plugs. But it was always absent from the healthy cicadas. It was absent from the other Massospora plugs. So why was it so low? Why was recovery so low? Well, think about the active kind of period of these compounds. Whether you've recreationally had magic mushrooms, or you've taken Adderall for ADD or ADHD. I have ADHD, and I take a stimulant for that. So I know that I could take it in the morning and have it be effective immediately. And it's very fast acting, but it also has a very short half-life. And that could explain why after a certain point, you're not picking it up in some of the plugs. The other thing I wanted to point out that I probably should have emphasized here is that we found it in the conidial plugs, and we found it in the stage 2 spores, but it was far less prevalent in those stage 2 spores. And that's important when thinking about, okay, what is the intended purpose of this compound? And some of this is a little speculative because, the only real way to test it would be to dose cicadas with the actual purified compound. But as a lot of you know, these things are regulated by the DEA, and can't just go around buying amphetamine and dosing cicadas and having fun like that. Not without some serious permit work. So really interesting that we found these things. And when you start to think about the effect that these compounds have, I mentioned ADHD. Bath salts are another synthetic amphetamine that were sold commercially until unfortunately someone had their face eaten off by another person. Awful story, but it kind of goes to show you that the wrong dosage of these compounds could have kind of some pretty nasty effects. Fortunately for us, back in the 60s and 70s, there was a lot of interesting studies done before the DEA really cracked down on the use and testing. And they basically showed that the similar doses that we were finding in the cicadas were enough to cause kind of increased aggressiveness in ants, and also altered feeding behavior in blowflies, appetite suppression, as well as increased aggression. If you're a fungus trying to get your mycologist host to continue to mate and fly around and engage in kind of healthy cicada activities as if nothing were wrong, as if a third of your body hadn't been replaced by a chalky fungal plug, Well, then an amphetamine would be a quick solution for that because it would keep you not thinking about food and maybe keep you focused on being kind of aggressive and maybe continuing to try to mate. I put this here just as kind of a fun thing. I put this on Twitter for those of you that follow me there. But the DEA sent me a letter after I kind of self-reported this discovery because I thought if we're going to publish this any journal, they may say, "Did you have the permits to study these?" And of course, it was inadvertent. It was unintended. And the DEA basically says, "Well, look, you're not trying to isolate it. The levels that you're finding are pretty damn low. Don't worry about it. Just keep us in the loop." So it's a cool keepsake to have. But this was April of 2018, so you can see how long we've been working on this. But in terms of what evidence is provided, well, the compounds were present in independently sampled broods of infected cicadas, but never from healthy cicadas. And the known activities of cathinone, for example, explains maybe some of this focused effort, improved endurance. Well, I like to call it prolonged wakefulness. If you had a sword stabbed into your stomach and a giant fungal mass erupted out, you'd probably not be whistling, doing cartwheels down the street. Yet these cicadas continue to keep on keeping on as if nothing were wrong. And amphetamine would certainly provide that kind of prolonged wakefulness. But the hypersexualization is a little more complicated, and we're not sure that the activity level effects of the amphetamine can explain that by itself. So, this provides a plausible hypothesis for behavioral manipulation, but we don't think that it's the only explanation, nor are we denying the fact that we could be completely wrong, and it's just like a really wild discovery that's unrelated. So obviously the next thing that we do as mycologists and fungal biologists and geneticists is we say, well, why don't you just look for the evidence in the genomes of these fungi? Well, again, I just want to reiterate that this is it's an obligate biotroph and it can't be cultured. So anything we're doing with it, any studies, all the studies we published have been off of harvested plugs off the back end of infected individuals that were collected from the field. We generated some Illumina high-seq data early on for both Massospora cicadina and Massospora levospora. And these genome sizes were they were awful. They were super fragmented. And you'll find out why here in just a short while. But the Busco scores to indicate the completeness were just super low. So we were just getting super low coverage and not assembling well. But we could still do something with that data. So we basically took the known, the biosynthetic pathways for psilocybin and cathinone, which were spelled out from their respective hosts. And we looked for candidate genes for those biosynthetic genes in that cluster. And what we found basically was we found candidates, except all but one step. For cathinone, we couldn't find evidence of a phenylalanine ammonia lyase. But all the other steps, or at least candidates for all the other steps. We looked for evidence of coordinated regulation for shared motifs, and this was some collaborative work I did with Emil Gluck-Thaler and Jason Slott at Ohio State. But again, no smoking gun, but some evidence of some of the biosynthetic pathway clusters, and then obviously we have the evidence where we actually detected the compounds only from fungus infected individuals. But it was clear that if we were ever going to do anything beyond that, we needed a better genomic resources. So in 2019, this is 3 years after we started studying it, an opportunity popped up in Southwest PA. We were able to sample and we were able to collect hundreds of individuals. And that was a real great opportunity to do that. And that resulted in the largest fungal genome ever. Massospora cicadina is a 1.5 gigabase genome. We published this, or at least a draft of the genome in the Microbial Resources Announcements in 2022. And this is, it's a huge genome, and it aligns pretty well with other members of the Entomopterales. Entomoptera musci, it's just as big. And one of the reasons it was so fragmented is because it's so fragmented. It's full of transposable elements. About 92% of the genome are these repetitive elements. So the other thing that was crazy is, despite being perhaps the largest fungal genome known as of today, it had perhaps some of the smallest number of predicted genes, only 7,500, compared to Entomophaga mymyga, which has some 28,000. And this is a very closely related genus to Massospora. So despite being the large genome, some of the fewest genes, does that represent kind of its evolutionary dead end that it's kind of gotten itself into? To be determined. But we were able to generate this improved assembly, and really this is just kind of a comparison between that Michigan strain. So we did improve it, and that provided a good genomic resource that we can then move forward. I told you that we can only really harvest spores off the back end of these cicadas, and of course, it's an ecosystem. There's bacteria in there. There's all kinds of things, and we're able to do and publish some metagenome assembled genomes for these bacterial partners or bacterial associates or community members that were kind of dwelling in these fungal plugs, and show that there are some unique fungi or unique bacteria in there. So I get asked the question a lot: can I eat it? Can I eat those plugs? Will they make me high? Can I replace my, there's an Adderall shortage. Can I eat a bunch of Massospora? There's a bunch of funky bacteria in there. And I'll tell you what, if you smelled half of the cicadas that came to me in the mail, the putrefaction and the decay, you do not want to put those things in your mouth. But in having this new genome, we were able to do basically an enhanced look at the kind of candidate genes for the biosynthetic gene cluster. And again, basically supported the earlier findings that we didn't have that first step, the ammonia lyase thing. We looked at some of the bacterial partners in there to see if they could fill that role, and sure enough, there are some candidates. So this could be kind of a coordinated effort between bacterial partners and fungal partners and hosts. Again, it's very complicated because we can't culture them, and we have to ask questions from wild type plugs, which are full of a lot of mystery, and there's a lot of confounding variables. But really interesting to look at it and kind of get similar results even with improved genomes. I don't know why this, it looks like part of this was one of my pictures came out, but basically these are, evolutionary oddballs, and they present some challenges because of all of these transposable elements. It's really hard to look for biosynthetic gene clusters because early diverging fungi outside the dicaria, outside of Ascomycota and Basidiomycota, don't really have the same kind of gene clustering that we see in there. And therefore, any tools we have to look for those clusters are based on, and built on looking at Asco and Basidiomycota. So that presents some real challenges. One of the things that we could start to think about is, okay, well, if fungi could be domesticated by cicadas like Ophiocordyceps, could Massospora be domesticated? And are Massospora domesticated inside their nymphs, and then come out? They need a way out in the adult stage. That's a question that we like to ask, and we're hoping to answer. Of course, as we move forward in time, new specimens show up on iNaturalist, and I hunt all the people down and I say, could we get that specimen? And we do. And I have a former grad student who is a perpetual through hiker who always collects me more and more cicadas out west. We've recently dug into herbarium specimens, which has been super exciting. And we tried to generate some low coverage, short read genomic resources for some of these type specimens and early specimens, not all types, but some of the early specimens that are in Cornell's fungal herbarium. And we tried this out. We tried some assembly, and actually we got usable sequences from 9 out of the 11 specimens, including 2 of those cup specimens, Massospora spinosa and Massospora perai. And we basically generated low coverage genomes for just about all the Massospora species, as well as 2 arthrophaga, which is kind of a millipede associated entomophthalales that's closely related. We were able to generate more spore data from the specimens and show that Spinosa had some unique spores. This was really interesting. When you go back, we go back, look at this. The genome assembly was 375 million base pairs. That's a quarter the size of the cicadina genome. So a much smaller genome. And we felt like the assembly was pretty good on that. And again, we're continuing to generate morphology data because that's just what we like to do, is kind of go down that road and kind of run these things in parallel so that we generate really robust, comprehensive, and complementary data sets. So we're able to put together like a we pulled some of the ribosomal sequences out of those genome sequences, and then we're able to put together like just some quick and messy phylogenies. And sure enough, interestingly enough, is that the Massospora perai actually group with Levospora and Platypedia, and all these Massospora seem to be one species, and they're all on host in the subfamily Tibicinae, which is a cicada subfamily. But you can see that Spinosa is kind of sister here to Dictyroprocta and form a clade here with pretty strong bootstrap. This is only a likelihood tree, and we have a more robust tree, but this is just for visual sake to show that we're kind of resolving the placement of these things. What was really interesting is that as we started to look back at the phylogenies for the cicadas, we realized that there seemed to be what looked like evidence of the fancy term for it is parallel cladogenesis, but it's kind of co-speciation. As cicadas move forward in evolutionary time from the beginning, so too did Massospora. And you can see that what complicates this a little bit is that Massospora levospora is a multi-genus cicada pathogen, whereas these others tend to have like one genus. And we're missing a lot of data from in the middle, but you can clearly see that there's some kind of mirroring going on, suggestive that this speciation happened together over evolutionary time. And there's formal ways to test that, but this is just a visual, thing to get you thinking about these things. What was even more interesting is that we could never get ITS to work. This is the barcoding sequence for Massospora. There's no sequences in GenBank. We couldn't generate any despite trying to find this. It turned out one of the reasons is that ITS is hypervariable. ITS2 is hypervariable in Massospora. Look at this. ITS2 is 2,339 base pairs long, and that's between 5.8S and 28S. And down here, it's 312 base pairs. That is a crazy amount of real estate to cover in just a single genus. So an incredible amount of length polymorphism, which probably explains why we're not able to get sequences. The other thing is that there is definitely some SNPs, some single nucleotide polymorphisms in the primer binding sites for ITS4, which is closer over here to the 28S region, that don't work for Massospora. And we were able to leverage these genomes to basically align this and develop primer sets. So we now have a primer set for Massospora that'll work on soil, that'll work on, environmental DNA, as well as Massospora. And we vetted it, and I've tested it already on the brood from down south, brood 19, and it works very well. So how could these broods help us fill in the gaps? Well, currently, Massospora is the cicadina is the largest genome, yet it only has 25% of the predicted genes of its closest relative. we are coming out specifically to collect for RNA sequencing so we can get some transcriptomic data to better annotate our genome from the 17-year cicada genome, and try to understand what genes are actively turned on, and if any of those are related to cathinone production, and try to understand some of those things. So we want to generate some resources in that regard. We're able to also maybe start to fill in the gaps about the life cycle. One of the biggest questions I have, and maybe leveraging some people on this call in the not too distant future, is if we can get people to send us eggs or nymphs before they fall out of the branches, can we run these targeted primer sets on them and ask whether or not we detect Massospora in first instar nymphs, in surface disinfested first instar nymphs, to indicate they are infected. We have about 250 plus observations of Massospora right now on iNaturalist. iNaturalist is a great thing. Cicada Safari app, there's other great opportunities to track these things, but we've made contact. I already have 128 specimens in the lab. That's from brood 19. We've already sequenced a number of them. I've done the morphology of them. When we go fast and hard. Like, we are a little bit obsessive. Sorry, I don't know why that pause sharing. So we want to sample more, and we have more coming, but we really want to get some good 17-year sampling. So, and I have some people already collecting around Chicago, but we're hoping to capture a number of things. So we're continuing to sequence, look at spores, characterize things, and see what's actually happening. We want to sample across every brood, across every species, and we have probably, if this was a United Way fundraising bar, I'd say we're at the 87%. We have from most broods and most cicada species, but there's some stragglers, the Septendecula, the Tridecula. Some of those are challenging. I want you to think about this fungus the way that you think about rust fungi. Rust fungi too have some of the large genomes in the kingdom. They're the only other group of fungi that come close. You can see them kind of here in the colored here. And here's the Entomophthorales over here. They're also obligate biotropes, and they can tell us a lot about things happening. And I'm starting to think more and more about rusts as I start to try to decipher what's happening underground. When we think about heteroecious macrocyclic rusts, that is 5 spore stage, 2 host rusts. We think about the 5 spore stages, the 2 spore stages that disseminate to the alternate or the primary host. And I think there may be something like that going on, where instead of having 2 hosts, we have 2 different life stages. I think there may be additional spore stages that happen underground in nymphs that we can't see. But those particular spore stages are non-lethal stages. And they may actually be beneficial or neutral. Now, I have some limited evidence because we're starting to see a 3rd spore stage that I hadn't seen before in some of the plugs that I've received. I need more data on that, but it's a really exciting time to be studying this fungus. But how can you continue to persist when 90% of your genome is not you? It's just repetitive elements. At what percent are you no longer you? And at what point do you become an evolutionary dead end where you can no longer function? Will Massospora drive itself into extinction? I don't know, but it's a real interesting system to think about. And I've taken a lot of your time. I hope you've enjoyed it.
Thank you so much, Matt. That was amazing. We already have a couple questions in the chat here that, yeah, maybe I can read off to you.
Yeah, I can pull up the chat too if you want.
Oh, yeah.
I stopped sharing just to make it easier. And I like to see people's faces. I mean, if they want to, I guess if they want to share their face.
We're all in our post-work outfits. No one wants to.
Oh yeah, okay, that's right.
Okay, here.
I think I missed how is the amphetamine showing up? So there is a naturally occurring amphetamine called cathinone, and it occurs in cat plant in the Horn of Africa. It's found in Yemen and in places like that, and it's chewed socially by men in those societies as a stimulant, and it's often around intense discussion. But we found evidence of that naturally occurring amphetamine in a fungus, not in a plant, and far removed geographically, which raises a question: does cat plant have a hidden fungal partner that's helping to produce that compound? That's what I like to ask. And I think that's the case. And I've tried to knock it out because you can work with some of the closely related plants from the Southwest that produce these same kinds of compounds. I'm doing that on the side, but there's only so much you can do. May have reached an evolution. Well, the evolutionary dead end point was when you have so many repetitive sequences and so few functional genes at some point, like, are you not going to have everything you need to survive? And it just seems like it's like this hyperinflated, I don't know, it's like the equivalent of being a bodybuilder and finding out there's only like a paper thin skeleton underneath it. It's just like there's this facade that it's like there's something big going on, but like there's very, very little function. But it continues to persist. So I mean, I haven't figured it out completely.
And in keeping with that question, I'm gonna ask them out just so that we record these and put them on YouTube later. So that it's not what was the question they're answering. We'll have that. So this person wanted to know about the one that you were tackling there. Can you explain how the fungus may have reached an evolutionary dead end? And I was wondering, like, maybe to supplement the info you just gave, I really like the bodybuilder analogy. For folks who have never heard the term before, what even is an evolutionary dead end? What's meant by that?
Okay, well, we know that organisms, including fungi, will accumulate mutations over time. And some of those mutations lead to them having an advantage in a given environment, and some lead to them being less fit or having a less of an advantage. And in some cases, fungi are also complicated by the fact that they have some unique reproductive quirks. There's parasexuality, there's unisexuality, there's secondary homothalism. There's all these kinds of interesting mating strategies, some of which are partially defunct, some of which are fully capable. So as much as you think, well, I mean, if you accumulate mutations and you're less competitive, maybe through recombination that you can get back to a place where you can survive. But a lot of these obligate biotrophs, we're not seeing evidence of sexual recombination. So these transposable elements can serve that purpose and kind of diversifying the genetics of these organisms. But it's complicated, and it's so complicated that I don't know that I can articulate it well, but it's really hard to reverse after going down a path with a cicada partner for 100 million years and accumulating all this trash in your genome, and then still having what you need to function.
Awesome. Let's see. I've noticed a couple people have maybe started to drop off. They have another thing. We can keep talking, but one question that I think we should definitely answer ASAP, if any folks do need to head out of here, is if they come across Massospora infected cicada, how should they save a sample if they want to like get it for you or something like that? What do they do?
Well, we have a lot of people collecting. I think the best thing you can do is upload an observation to a community science platform like iNaturalist. I mean, there's Mushroom Observer. There's other ways to communicate that. You can contact me directly via email or on social media. But I think it benefits more than just me if you upload that observation to iNaturalist, because we could use that data set. And then you don't even have to tell me. I'll know it because we have email notifications of every Massospora that comes in. So I would say that's good strategy. If you have a hotspot where you're seeing a lot of them, just reach out. You can find me on my email on my faculty page at WVU. You can find me on Twitter. I'm very active there. So or you can reach out to Kate, and Kate could get me the information. So like, even if you don't know my contact, you can reach out to leadership at the association. They can get that information back to me. But I think
Absolutely.
the thing you can do that will benefit you directly is to observe cicadas as they emerge, because this is the only place in the world that we have periodical cicadas. And although they scream and we're mad at them, it really is a thing of beauty, like a biological phenomenon that not everybody gets to see. And the fungus itself is a real interesting biological phenomenon. So, don't eat it, observe it, photograph it. You're never going to get sick from taking a photograph of a fungus. So enjoy it and report it.
Excellent. We had another person ask a question
Yeah.
about why do cicadas, or excuse me, what, I can't read. What do cicadas and Massospora ultimately do for the environment?
Well, I think to answer that, we have to think about nativity status. We know that periodical cicadas are native to North America. We know that the fungus is native. Although there's hot spots where we can see infections upwards of 20% or more, across the larger landscape, infections are pretty low. The incidence of infection. So it's just kind of managing. These cicada emergence, these broods they're super spreader events, right? Like there's going to be a lot of disease. And Massospora is just one of the players that's going around saying, hey, let's take advantage of all these cicadas. And there's plenty, there's so many cicadas that every bird and snake could fill their bellies full. And, and then there's a boom in population growth in those years. And there's still enough that the cicadas can, lay enough eggs that the next generation survives. And there's still enough that Massospora can infect. So, it's just, there's a lot of things that have kind of jumped on the cicada bandwagon for good reason. It's just like a mass. It's an opportunity to persist.
Yeah. And I think I'll kind of piggyback onto that with, I've asked a similar question to that many times in my work as a science writer. And it kind of presupposes that there's a reason for anything. Most of biology works because it happened and it didn't die, so it's going to keep doing that. Cicadas, they're not, they're not setting out to be like, well, there's going to be a real bumper crop of squirrels and birds this year because they're going to eat some of us. They're just trying to get out there and mate, like the rest of us. And some of Massospora, they're not, I hear this often when people talk about why are parasites in general important. It'll be like, oh, they're good for population control of other things like that. The parasites aren't, consciously in any way saying, oh, we're just tamping down on the numbers of grackles or sloths or whatever animal they're parasitizing. They're all just, trying to get a fair shake out here. It's
Yeah.
nature is anarchy.
I think the thing I think about is that some of the more speculative slides I had at the end about comparing it to rust fungi, I suppose that there may be some kind of advantage it may give, or like even let's just like throw out some wild speculation, some protective effects to the nymphs. There's a lot of different pathogens that could invade these very fragile, early instar nymphs. And obviously they've survived for a long time, but like maybe Massospora is able to confer some kind of advantage if they are in fact infecting them on the way down, which makes way more sense than them infecting on the way up.
Yeah. Also, I think John Cooley, where's he? Is he at UConn?
Yeah.
Yeah. He's another scientist who does a lot of research with cicadas and
Oh yeah,
into
he's
Massospora.
published. He's been co-author on several of our papers.
Yeah, yeah. I know one hypothesis he's had as to, is there any potential benefit that Massospora confers on the cicadas? Because it seems like the cicadas are getting a really raw deal here. But, one of his theories is that since the Massospora is producing these amphetamines and psilocybin and things like that. His theory is that if you're a young bird out looking for a cicada meal and you eat a cicada that's infected with Massospora, you may have a really bad trip, and that bird is going to have the worst day of its little bird life, and it will say, lesson learned, I'm not fucking with those or I'm not doing anything with those cicadas anymore.
Well,
And that there's potentially a population-wide benefit.
there's some anecdotal evidence, and again, a lot of this is we just need more natural history observations. Is that infected cicadas are just kind of like more wild flyers, and maybe it's good for a little bit of range expansion if you can get infections that still allow you to reproduce. That is, say a female that is mated and ready to oviposit eggs gets infected, but not enough to make her like the viability to affect the viability of the offspring, then maybe she can expand the footprint of that otherwise, kind of tight geographic kind of overlay of that brood. Because she's like, you know what? I'm on amphetamines. I got some eggs to lay. I feel pretty good. I think I can go over that football hill.
Let's go for it. Yeah, I mean, and I think it's true also that for lots of parasites, when you come with one, you find an individual who has been overtaken by the parasites, like clearly something has gone wrong immune system wise for them, but that most healthy squirrels, chipmunks, ants, whatever that you see out there have some parasites, and that, on a low level, they're not bad, and it possibly even beneficial in ways that we're still learning.
Yeah, and it is
Matt
possible
Nelson
that
says he needs to run.
yeah, it's possible that
5 minutes.
way more cicadas have Massospora infections, except the load, like you said, the inoculum load might be too low to actually lead to this outward symptoms.
Yeah, that like to your point, that maybe for most of them it's like, oh, I'm feeling a little perky. I'm gonna go spread a little farther, as opposed to like my entire abdomen is falling off. Um, let's see. Somebody else asked, how is an amphetamine showing up? Is it naturally occurring somewhere in the world? The psilocybin makes sense, but I don't understand how amphetamine got into the picture.
Yeah, I mentioned that, but I'll say it again because I think it's interesting enough. Most amphetamines that are produced, including the ones we use for ADHD drugs, are synthetic amphetamines. But cathinone is a naturally occurring amphetamine that occurs in a plant called cat plant, a KHAT. And Catha sinensis is the Latin binomial. And they're closely related to ephedra plants, like the ephedrine diet drugs that they had in the 70s that made you lose a lot of weight. Yeah, Adderall has that same effect because they're stimulants and they just suppress the hell out of your appetite. But the question for me that immediately arises is that in the biosynthetic pathway for cathinone, we have some of the compounds that are found in ephedra plants in the Southwest, as well as cathinone from cat plant. And it makes me think that maybe some of these early diverging fungi too are endophytes of some of these weird drug plants. And we just can't culture them out because they're unculturable and they're actually like cryptic fungal partners of these plants. And explains how there could be these what seems like far-fetched independent evolutions when in reality everything's fungi because everything always was fungi.
Amen. We have a question asking, has Massospora been cultured on plates?
We can get it to germinate from both the stage 1 and stage 2 spore stage. And we've grown it on there's a lot of like weird media that the weird substrates that insect pathologists have used, egg yolk agar. Like we've used all like breakfast agar. It's like, skim milk plus egg yolk, autoclaved, of course, but like none of it. Grace's media, which is supposed to mimic hemolymph of insects, none of it will keep it growing. So it's probably been coerced into germinating, but not budded or anything like that.
Just wants the bugs.
The way the way that they were able to sequence the genome, chromosome level genome for Entomoptera montmyga, was they got blastospores. They basically were able to kind of keep it proliferating in liquid culture as blastospores, and we could try to do that. Although it's still highly artificial. It's not like growing it. Well, I mean, I guess growing it on solid media would be artificial too, but.
Excellent. This person asked, could some of the different bacterial partnerships have an effect on the signaling or production of those fun secondary metabolites?
Certainly. And we've thought about that because what's really interesting is that when we sequenced the genome of for Massospora, we were unable to detect the bacterial partners. And that's where we came up with this hypothesis that maybe the fungus was acting in that role. But how would it get in? How would the fungus get in and invade detection if it somehow uses some of these bacterial signals, or there's got to be interactions, three-way or more interactions between the bacterial partners, the fungal parasites, and the host, and who's contributing what when is the biggest mystery of all.
Yeah. Someone wanted to know what percentage of cicadas have this fungus? I know you said it varies a little.
Yeah, I've sampled in some hotspots where infection incidence is 25-30%, like where we got all the specimens for genome sequencing. And I've had one of my collectors in South Carolina, he said it was 30% on one species there, but I would say below 5% averaged across all the landscape.
Yeah. Let's see. We've got a question here that is actually a lot of these are more comments than questions, just saying that people really enjoyed this. Here's a question. Any idea how long the spores persist in the soil? Do infection rates in an area increase each cicada emergence?
Great question. The field studies that have been done are not really super convincing. The science wasn't shoddy. It's just the understanding of the fungus was incomplete at the time the experiments were put in. These resting spores are very thick-walled. I have no hesitation to say that they can persist for years in soil. But the fact that soil is turned over so much by earthworms and invertebrates and all that, the likelihood of those spores actually encountering cicadas go real deep in the first couple of years, especially in the cold. They'll be meters down. Like Massospora spore is going to make its way down there? No. And in fact, we don't, like, our original hypothesis was that they came in the months leading up to emergence in year 17 or 13. It was in those vertical emergence chambers as they were waiting for the soil to reach 64 degrees. We thought that's when they would encounter the spores and get infected. But I'm just not convinced. I'm not convinced that the spores infect on the way up. So I think that they're infecting on the way down.
Cool. Anybody else have anything? Matt, as someone who talks about this all the time, is there anything that you wish people would ask, that like a fun fact that you want to share and never have a chance to?
Fun fact is I've never received a dime of funding to do this work. This is all like side work that I've done. We've published 5 papers on it. And I could get a grant on it, but I'd rather just keep working. I'd rather just keep driving out places and collecting them, because this is really like my passion project.
That's right. As long as it's on your terms. I was ready to be like, that's not a fun fact at all, Matt.
No,
We've got
it's
a
fun
question.
because it's like, it's one of the real productive areas of my lab, yet it's not fun to do.
Let's see. We got a question here. How dangerous is it if you've recently licked a Massospora infected cicada? Asking for a friend.
I licked 6 right before this call, just to see what happened. No, the amount per plug is biologically significant for a cicada, but I can't see everybody because everybody's screens are off, but I guarantee you everyone is heavier than a cicada here.
This is why they don't want to turn their screens on. They're all cicadas.
So you would have to eat dozens of plugs to have an effect. And you'd already be so sick from all the bacteria. And, I've seen fungal gnats. I've seen other insects laying eggs in dead cicadas with plugs. I've seen slugs just like licking and rubbing their bodies all over those fungal plugs. Do not put them in your mouth, ever. Yeah.
Excellent.
But so one thing I'd love for you all to keep your eyes open for is that I mentioned that there's these green cicadas that occur in the mid to late summer called dog day cicadas, and they're far less abundant. You see them more, or you hear them more than you see them. But a Massospora infected dog day cicada, I've seen photographic evidence, legit photographic evidence, because it was inadvertent, of one, and I would pay real serious money for it, probably upwards of $250 if someone were to come up with one.
Well, hot dog, I've got myself a mission come August. And I guess that does kind of lead to, like, I know with things like Ophiocordyceps, it's incredibly species specific. Like it's almost a 1-to-1, I think, of
Yeah,
like, this is one kind of Ophiocordyceps, one species of
right.
ant. Do we have a sense? I mean, at the beginning you were talking a little bit about the the spread of different types of Massospora, and I admit I was just frantically adding attendees to the Zoom and missed a
Yeah,
little.
no, no. So
Like,
what I
does
said
the
is
same Massospora cicadina infect Magicicadus cassini and Septum decim and all the others?
right. That was a question. It was actually a lingering question because what all these studies we've ever done, we only had 2 specimens of 13-year cicadas. Everything else was 17-year cicadas. So now that I have 128 in the lab, I've already sequenced a bunch of them, and I can tell you that those ones I sequenced are a match to what we have. So right now, cicadina is one species. There's other Massospora that infect other cicadas. So to broadly answer your question, the general rule for Massospora is one species of Massospora to one genus of cicadas, except for Massospora levospora, which infects multiple genera in the same subfamily.
Huh. Cool. Another person wrote in, what's the best way for someone to send you a cicada? The best, I think it's mostly take a picture and upload it to iNaturalist.
Well, if they want to send me, if they want to actually, they collected it. If you have a good specimen, if I see a picture of it and I want it, I'll send you a tube so you can send it to me, because we have some 15 male falcon tubes that are like a little bit more durable for the male. I've had people roll them in toilet paper and send them in a bag, and they come like flattened and like looking just like a powdery mess. So I'd rather send you some tubes, but that would require you to touch base with me, send a picture. It's better if you put it on an iNaturalist, then you can send me the link and we can go from there. It may be a spot I have 473 and I don't need any more. But I also appreciate your interest and I want to hear about it.
Cool. All righty. Well, it's getting to be 9 p.m. I think probably time to call it. But thank you so much, everybody, for joining us. Thank you, Matt. This was so fascinating, so fun. Yeah, we'll be sure to be in touch as you come to Chicagoland later this week. And let you know any good cicada hotspots. And
Yeah.
thanks for joining us.
Yeah, for sure. I'll probably CC you on, and I saw Matt was on earlier. I don't know if he's still on.
He left because he's a traitor. Yes. He.
Okay. I will CC him because he had reached out to me and just to
Yes.
get yeah, to everybody involved. But I really appreciate your willingness to have me talk, and thank you for dealing with my ADHD procrastination that took forever to get a title and a commitment.
All good. This was very, very fun. Thanks so much, and hope to see you soon. And thank you everyone for coming. Join IMA if you're not already a member. Go to Liz's talk next week, and we'll see you in the woods.
See ya. Bye.
Bye.
Thank you.
Thank you.